we have learned the signal hypothesis by which we know that there is a signal which is associated with a protein that is targeted to the ER as a soluble protein. Now, how actually the scientists actually know the protein has extruded inside a microsome? So let's take a look at that. This is a microsome, and on top of that, suppose this is the large subunit of the ribosome, the small subunit of the ribosome, and you have the mRNA right here, the mRNA, and the protein is translocated inside, which does have the signal peptide. So right here is the signal peptide. So how do we know that this protein is extruded inside the microsome? So for this, some elegant experiments can be done. So what you do, that you do two sets of experiment. So in one experiment, and you are doing this in vitro translation in the test tube, and again, this is exactly the same. So this is in vitro translation, reconstitution. So you add all the material that is needed, all the tRNAs, all containing all your amino acids, all your buffers, all your enzymes, your ribosomes, your mRNA, all the material in the test tube. And what you do, that there are two different test tubes you take, and one is minus the microsome. So one is a minus microsome. That means that you do not add any microsome to that mixture. Another was with the plus microsome. That means that you add the microsome to the mixture and let the experiment actually happen. And when you do this experiment, we have seen that in the Gunther Blobel's experiment, plus microsome does have a smaller size of the protein. We know that. So here, instead of having the microsome minus and plus, we are modifying this experiment just a little bit. This is essentially the Gunther Blobel's experiment to understand whether you have the attached signal peptide or not, and the difference in here. Here we have two different plus and the minus microsomes. When you do, you add in one of them, you add the microsome along with others. So on the other hand, what you do, so this is, you are adding microsomes while translation is going on. That is your first experiment going on. So in the first one, this is what is your experiment. Microsome added along with other constituents in an in vitro translation reconstitution while the translation is going on. On the other hand, what happens, the second tube that you use, you do the translation minus microsome, just like the previous experiment like Gunther Blobel, and then after the reaction or the translation going on, you add the microsome. So this is what we called as that you have added the microsome, post-translationally. So after the translation, this is essentially, in one word, if we just write that what you are doing here, that you are adding the microsome co-translationally. You are adding the microsome co-translationally. Here you are adding the microsome post-translationally, after the reaction actually took place. So this is the only difference. So essentially, how the picture will look like if you do these two experiments right here. So what it was found, that right here is the microsome, and all the proteins right here are present inside the microsome. When the microsomes were added co-translationally. Unfortunately, or well, the other type, not really unfortunately, but by the experiment, when the microsomes were added post-translationally, it was found these are the microsomes, and proteins have not extruded inside the microsome. They remained outside. So how exactly you can identify this is possible? So again, remember that we can do this experiment the same way that when we do this experiment in vitro constitution, we again use 35S methionine as a radioactive source to label the protein. We will use an antibody which is attached to an agarose bead to do the immunoprecipitation reaction to do this. But before adding that agarose bead, we do one experiment here. To this, we add also essentially a protease. We add a protease which should digest the proteins. Here also we add some protease. That should digest the protein as well. So after adding the protease, we add this antibody and we do the immunoprecipitation reaction, and after that we run a gel. So how this gel now should look like? So if we just have this gel, and here are the two different things. Here we are talking about that here you have added the microsomes, co-trans, and the other one is post-trans. So microsomes. So these are the two different conditions that what you have, two different lanes on the SDS page. You are running an SDS page, SDS polyacrylamide gel electrophoresis. After that, again, just repeating in short that you have co-translational added microsome, post-translationally added microsomes. After that, you have treated both of them with a protease, followed by an antibody agarose bead immunoprecipitation reaction to isolate this particular protein against which you have that antibody, and then you run the gel. And when you take after running the gel, when you take an autoradiograph, as it is radioactively labeled, you will see the signs in the autoradiograph. So when you see the post-translational microsome added, you find your protein. Right here is your protein band. And your post-translational modification just shows some radioactivity essentially at the bottom of the gel. There is nothing. So essentially what it tells you. So this experiment tells you that the protein has been extruded into the microsome. How? The reason for this, as you have added the protease after the reaction, only if the protein is inside the microsome, protected by the membrane of the membrane, it is protected from the protease. Protease does not have any effect on that because the protease cannot penetrate through the membrane of the microsomes. On the other hand, when you added the protease, this is the mixture where you have added the microsome post-translationally. So the protein did not end up going inside in here. They did not. They remained outside the microsome, and they were all digested by the protease. So when you immunoprecipitated them, you did not get anything. You get some just random fragments and radioactivity, which is at the bottom of the gel. You did not get the protein. So this tells us the co-translationally, when the microsomes are added, the proteins are essentially getting inside the microsomes. When microsomes are added post-translationally, the proteins do not translocate inside the microsome. And with this elegant experiment, we can actually tell whether the proteins are inside the microsome or outside the microsome. Now, we can modify this experiment a little bit as well, where we can again, how to confirm that really this is inside that membrane. So we can do one thing right here. We can take this co-translation, this microsomal fraction, and then right here, and after that, what we can do, that we can take a test tube where we have added the protease for sure, but before adding the protease, we do something else. We add plus a detergent. We add a detergent. So after the detergent, we add a protease plus protease. So now when we run the gel back again, suppose this is just only that single lane, essentially what we will find that if we just modify this just a little bit, both are co-translational, and so both the lanes should look at co-translational translational, both of them. What one is minus detergent, so protease plus minus detergent. You do not have any detergent, means nothing, just only protease essentially. And this is protease plus detergent. So this is without any detergent, only protease. You are going to find the band right here where you found. But when you have added the detergent, you get the same result. So what does that mean? That the detergent made holes into this membrane, and after it made the hole into the membrane, the protease could enter into the lumen of the microsome and digested all the protein that was inside, and you do not get any protein. So this again tells us that, or confirms us doubly, that the proteins are indeed extruded inside the microsome.